Hey everybody, welcome to Anatomy 2. This is going to be our first lecture on the blood. My plan is to keep these lectures pretty concise and to the point. So with that in mind, let's dive right in and see where this thing takes us. So the first thing we need to ask ourselves is, what does blood do? And there's a variety of functions. There's a lot happening there. It's a little more complicated than just this red substance flowing through our veins. Blood is incredibly important as a unit of transport, all right? Blood carries a lot of things for us, oxygen being key amongst these. So we're going to be picking up oxygen in the lungs, carrying it around the body to whatever tissues are needing it. And then as we go through cellular respiration, we use that oxygen. And then we release, as a byproduct, we release carbon dioxide. So that carbon dioxide will then be picked up in the blood and carried back to the lungs to be released. So we pick up the oxygen, use the blood as a transport mechanism, and then carry that around to where it's used, then releasing carbon dioxide therein. We will also be picking up nutrients, all right? So nutrients are going to be picked up in your intestinal tract. They will be picked up there, taken around the body, again, to wherever they are needed, utilized, and then you can expect some sort of waste product, a byproduct of our cellular respiratory pathways that will then be picked up in the blood and then carried around to wherever they're going to be eliminated from. So the blood is the transit mechanism, okay? The blood is the transit mechanism. It's going to be picking things up, carrying them where they need to go to be utilized, and then picking up any waste products that are resulting afterwards. And we can't neglect the concept of hormones as well. So in our system, we sort of communicate in a couple ways. So internally, we can communicate via our nervous system, so sending messages from place to place via nerves. But our hormones are also very important signaling mechanisms. Again, think of the ways in which we regulate, for instance, our blood sugar. All right, we're going to release insulin. The insulin travels around the body and stimulates certain cells in our system in order to take up excess blood sugar. That insulin is traveling, as a hormone, it's traveling through our bloodstream. So our bloodstream is a medium through which things move to get from one place to the next. So our blood is an incredibly important transport mechanism. Two, our blood is very important for protection. Inside our system, we have the capacity to deal with damage. All right, so if you are in a car wreck or just generally get a cut or fall down and get a bruise, you will have damage to your cells. And our blood has inset mechanisms for clotting. They're going to be protective measures to allow you to prevent blood loss in the event of damage. And further, from infection. So our system, our blood certainly protects us from infection using white blood cells. So we have white blood cells that are going to be set up specifically to attack, for instance, bacteria, or to attack certain parasites, or attack virally infected cells. Our blood has mechanisms internally. We have white blood cells that will allow us to deal with certain problems. It is most certainly a protective mechanism. And then last but not least, regulation. Our blood is certainly going to play a role in regulating our temperature. You have to think of your blood as a means of conveying heat. Okay, it carries heat from one place to the next. If you're hot, your blood will carry heat to your skin surface. And the idea is that by carrying heat to the surface of your skin, it's going to allow that heat to escape from your body, thus bringing your core temperature down. By comparison, if you get very cold, your blood will remain in the core of your body. It doesn't go out to your extremities at nearly the rate. You'll feel this. When you get cold, where do you notice it first, right? Tips of the fingers, maybe your nose, your feet, cold feet, legendarily. All right. The idea is, and you've probably heard of the concept of frostbite, you can lose a finger or two and live as long as you keep heat in your core and in your brain. If you keep the brain and your organs working, you might survive. Whereas if your core temperature drops and you go hypothermic, you can die quickly. Okay, so blood conveys heat from place to place in your system. That is absolutely factual, simply a heat transporting mechanism. And then, of course, pH. So your blood has an inset, what's called a carbonic acid bicarbonate buffering system. I'm not going to go into a whole lot of details on this, but the idea is that your blood is a little bit on the basic side. Okay, so your blood's pH is slightly alkaline from about 7.35 to 7.45. You can get acidosis where the blood gets too acidic, for example, or the opposite of that, the other side. The idea is we want the blood to stay in this neighborhood so it has appropriate oxygen carrying capacities and it's able to move things around when necessary. Okay, it's functional in this pH range. What else do I want to say? And regulation of fluid volume. So your blood is obviously mostly water. Okay, your blood is a lot of water, and by carrying that water from place to place to the kidneys, as an example, we have the capacity to regulate the amount of fluid in the body, and that can in turn play games with blood's viscosity. It can play around with your blood's pressure, a whole variety of things. So your blood is certainly part of a regulatory function. Your blood has a variety of characteristics. It's hot. Okay, your blood's very warm. A friend of mine that was a surgeon, he said the biggest surprise he ever had was the first time he got his hands into another person. Okay, he said it was almost uncomfortably hot, like it hurt. Okay, your internal structures, your core temperature is quite high, and it's hot to the touch. So your blood is most certainly going to be hot as well. It's got a sticky feel to it because it's got a lot of fiber inside of it, believe it or not. And we'll be talking more about what the role of that fiber is as we progress. It's got a salty flavor, all metallic to some degree. And we're all aware of this. You've bitten your tongue before, or bitten your lip, or had a busted something. Or maybe you're a kid and you hurt your finger and put it in your mouth. Gives it that metallic taste because there is a lot of a lot of metal ion in your blood. Again, you have a lot of iron in your blood. It gives it this metallic taste. It's got a red coloration to it, and it kind of depends upon the level of oxygen. Blood cells, red blood cells that are low in oxygen get this deep crimson color, and then red blood cells that are highly oxic, that contain a lot of oxygen, will have a really nice bright red color. Okay, so it depends upon the amount of oxygen that's there. I get questions all the time. It's like, what? I thought my blood, thought it was low oxygen. I thought it was green. Like I can look at my vessels and they look green. No, this is just a side effect of how light is refracted. I assure you the blood in your veins is red. Okay. What else we have? And we talked about the pH, so we're good on that. All right, so that is the general idea of blood and where we are going. So let's go here and talk about the composition. So what we will do, a good way to identify if someone has some sort of health issues to take a urine sample and a blood sample. With those two things, we can pretty well diagnose most major problems. What we'll do is you will pull blood from the body, spin it down in a centrifuge, and you get an output that looks something like this. And what you find is that blood is about 55% plasma, about 45% red blood cells, and then less than 1% what's oftentimes referred to as a buffy coat. Okay, buffy coat. This would be white blood cells. Okay, white blood cells and platelets for that matter. All right, now with that in mind, blood is a liquid connective tissue, the only liquid connective tissue. Now, most of your connective tissues are quite solid because they have a lot of elastic fiber or a lot of collagen fiber. That is not the case for blood, but blood is still considered a connective tissue. It certainly connects one part of the body to the next. It comes from embryonic mesenchyme, so it's got the same embryonic pathway as other connective tissues in the body. And all connective tissues have to have the same basic structural components: cells, ground substance, and fiber. And blood certainly has cells, ground substance, and fiber. Let me find a good picture here. So there are most certainly cells here. Ground substance would be plasma, that clear background that you can see there. And the fibers would be related to that which leads to blood clotting, as an example. So there are certainly cells, ground substance, and fiber making blood a connective tissue. Good, good. Why does it still have fiber? Again, for clotting. These fibers are going to be dissolved into the plasma, and if you have damage to a vessel or what have you, they will come out of suspension and lead and assist, I should say, with that clotting effect. All right, fluid connective tissue, plasma, non-living fluid matrix, we've already established this. And very important here are the formed elements, okay? The formed elements, these are the living cells. These are the erythrocytes, the leukocytes, and the platelets. You need to be friends with these. Hi everybody, and we are back. A little change of venue, minor nuanced problems, but regardless, we are back. And I believe we were talking about the components of blood. We had established that blood is in fact a connective tissue. We established that there's a non-living matrix, which is part and parcel with being a connective tissue. referred to as plasma that's within blood, and that plasma makes up the majority of the blood itself, about 55% of the whole blood content once it's spun down in a centrifuge. And then, of course, there are the formed elements. All right, the formed elements are the cells for our purposes. Okay, the cells are erythrocytes, which you'll also hear called red blood cells. You will also hear these called RBCs. They are erythrocytes, and they're going to be the cells that are primarily responsible for carrying oxygen in your tissues. There are leukocytes. Leukocytes will be white blood cells, or WBCs. You'll often see them called. Leukocytes help us fight off infections and deal with certain parasitic infections. You'll see there's a whole variety of them. When it comes to red blood cells, a red blood cell is a red blood cell is a red blood cell. But when it comes to leukocytes, white blood cells, there is a grand variety. Okay, a grand variety. And then last but not least are platelets. And platelets are basically just cellular components. Platelets form from a single large cell that bursts into a bunch of fragments, and those little fragments are in fact platelets. And you will find that they last a very short period of time as a result of this. All of these will have varying shelf lives, but platelets tend to last very short periods of time. All right, so let's see here. We have talked about spinning our blood down. We know that the top portion here is very light, kind of straw colored, is what they often call this. This is plasma. And then we need to talk about, let's see, we've established that the leukocytes in this buffy coat make up less than 1%, including platelets, and then the bottom part. Okay, the lower portion of this, which on this graphic is around 45%, that would be what's referred to commonly as a hematocrit. All right, that's the percentage of blood volume that is red blood cells, hematocrit. And what you'll find is that this varies between males and females. Okay, about 47% will be red blood cells in a male, and about 42% will be red blood cells in a female. And what you'll find is this is due to the effects of testosterone as a hormone augmenting the production of red blood cells. All right, so that's sort of how it works. All right, perfect. Let's go here. So let's talk a little bit about blood plasma. And again, this is the real thing. You can see this kind of yellowish, what they call straw color here. When it comes to plasma, the majority of it's water. Okay, the vast majority of plasma is just water, but there are other things in there as well. In fact, hundreds of other things in most cases, many, many other things. Lots of various nutrients that are going to be flowing in from the intestinal tract and moving around the body are going to be found in the plasma. Certain gases are found in higher concentrations in plasma. For instance, you know that red blood cells carry most of the oxygen, but most of the carbon dioxide is going to be flowing in as plasma, or in part through the plasma. Okay, it's not necessarily going to be attached to the red blood cells. Only, I think it's like 20% of the carbon dioxide carried in the blood moves via red blood cells, so very little by comparison. Hormones are going to be found in the plasma. That would also include antibodies. Wastes, like nitrogenous waste products, are going to flow through plasma. Certain ions, the ionic balance of your blood is very important. For instance, calcium has to be at certain percentages. This is going to be found in the plasma. A whole variety of things. And in fact, I've got a kind of an interesting little pie chart, if you will, here, outlining some of the major constituent parts of plasma. In particular, what we need to talk about are the plasma proteins. Okay, this is representative of the plasma proteins. And there are three major categories of these plasma proteins, and they pretty much exclusively arise from the liver. Okay, these are of liver origin, if you will. These are albumins, globulins of various types, and fibrinogen. Okay, albumins are important because they kind of play with the blood's osmolarity. By having an appropriate amount of these albumins in the blood, it sort of keeps the appropriate amount of water in the blood compared to in your extraneous tissues. If you're not aware, your blood has a constant give and take in terms of its fluid component in relation to the cells it's around and flowing near. In particular, your loose areolar connective tissue, if you're aware of what I'm talking about, there's an osmotic gradient between your loose areolar connective tissue and your blood, and the appropriate amount of albumin in the blood is very important for maintaining that osmotic balance. And in and of, sort of in concert with that osmolarity, this plays with the viscosity of the blood. And what you're going to find is, viscosity, by the way, the thickness, if you will, what you're going to find is the viscosity of the blood is very important. If the blood's too thin, if the blood's too thick, you have major problems that can result. Okay, major problems, and we'll be getting into those in a little while. So yes, albumin's very important, small but very abundant, make up the vast majority, more than half, and deal with the blood's osmolarity and thus its viscosity. Globulins, of which there is a whole variety. I mean, if you can pull this up on your screen and see it, there are a huge variety of different forms of globulins, but in particular importance for us are alpha and beta globulins that play a role in transport, specifically of ions through the bloodstream. And probably more important are gamma globulins, which are antibodies. Okay, gamma globulins are a very important part of your immune system, and boy, you're going to be hearing about these all throughout the semester. Okay, it's very important. And then last but not least, of our plasma proteins, again, of the protein found in the plasma, is fibrinogen. And fibrinogen is a major precursor, if you will, in the blood clotting process. So we said that there's a lot of dissolved fiber in the blood that leads to blood clotting. Fibrinogen plays a role here. This is a plasma protein. All right, and that takes us to the other formed elements here. So what we're going to do is we're going to go through and talk about erythrocytes, leukocytes, and platelets. And you can see these all here. Erythrocytes look like little pink donuts because they have a very traditional, what they call biconcave shape. Let's see. Yeah, let's just talk about it. We'll get to the rest of this in a second. Then we have our white blood cells, and again, these all look a little bit different from one another because they are a little bit different from one another. White blood cells can be highly variable. There's a lot of different types. And then, of course, there are platelets, and platelets are simply cellular fragments. As you look through a blood smear, like you'll be seeing a lot of them in this PowerPoint slide, you'll see some little bits of stuff kind of in the background. It almost looks like the slide's dirty. What that is, this debris, those are platelets. Okay, and they're going to be involved in the clotting process, as we described moments ago. Now, worthy of mention here is that red blood cells lack a nucleus. So when you look at the red blood cell and it looks like it's got a hole in the middle, that's because it's incredibly thin in the middle because it's shed its nucleus. And the lack of a nucleus in a red blood cell is going to play an important part in its lifespan. So red blood cells only live for like 120 days. Eventually they become non-functional for reasons we'll get at in a second, and they are eliminated from the body. They can't fix themselves when they have problems because they lack a nucleus which would provide the code for how to fix themselves. So no nucleus. And there's a reason they lack a nucleus, and we'll get there in a little while. But the point here is that white blood cells are the only real complete cells in your bloodstream. The red blood cells have shed their nucleus, and they lack most of their organelles for that matter. And platelets are just pieces of cells. So white blood cells are the only true cells that you have. And that's why some white blood cells can have a long lifespan. So most of the form elements survive in the blood only for a short period of time. Red blood cells, as I said, about 120 days. Platelets, only 8 to 10 days. And then white blood cells are highly variable. Highly variable. Sometimes white blood cells are made and they go directly into battle and die. Like immediately. They're made and they go die. They're made and they go die. Because you got to remember what these are going to be doing, right? They're going to be doing battle on your behalf to keep you from having bacterial or fungal or parasitic infections. They're going to be going and fighting against pathogens that want to hurt you. So they're literally going right into battle in a lot of cases and dying. And you're aware of this. If you have ever had an infection for one form or another, and then you develop what you might call a pustule, an area where there is a pus under the skin and that ruptures, and you clean it out, hit it with some peroxide, that pus is dead white blood cells. Okay, well, dead white blood cells and probably dead bacteria that have been in there duking it out to keep you safe. Okay, so white blood cells can be made and die immediately, and then you got white blood cells that are going to hang out in your tissues for decades. So it's very variable, very variable. Let's see what else we have to say. Okay, most blood cells, I would hasten to say just about every blood cell that we're going to talk about is going to be marrow in its origin. Okay, they're all going to originate in the bone marrow. And I put up a nice, I think this is a transmission, no, I think this is a scanning image. Don't quote me on that, but nice electron microscope image of a bone. You can see the bone's trabeculae pieces here. The red stuff in the middle, that is bone marrow, in particular red bone marrow. That's going to give rise to all these blood cells that we've just talked about. They're all coming from the bone marrow. Now, I want to point out one more thing here, and that is to note the size. Red blood cells are pretty darn small compared to white blood cells, mostly because they lack a nucleus. That's going to play a role here. But red blood cells are very small. Like, look at this. This is a lymphocyte. And lymphocytes are considered pretty small in the realm of white blood cells. And look at the size of that lymphocyte compared to the red blood cell. I mean, red blood cells are very small by comparison to the size of a white blood cell. And I'll give you a hint as to why that is. It deals with surface area. What do red blood cells do? What do white blood cells do? Okay, white blood cells are defending you against attackers. They need to be large. Red blood cells carry oxygen and have to exchange it very quickly in your tissues. The more surface area they have per their volume, the better. And that dictates they have to be small. We'll talk about more of that in just a second. And then again, the platelets are just cellular fragments. Like, look right there, there's a platelet. Platelets are going to be very small indeed because they're just pieces of cells. All right, let's go into a little bit more detail on red blood cells, the erythrocytes. And again, note that these are going to be non-nucleated, they lack organelles, they are very small. And again, as a result of losing their nucleus, they get this very traditional biconcave shape. Like when you look at these, they look like little pink donuts. You can see it in cross-section here. Very small, flattened biconcave discs. And the idea is that this vastly increases their surface area to volume ratio. The more surface area, the better, because the more surface area you have, the better you are at exchanging things quickly, like gases, for instance. So when you think about the actual exchange area where red blood cells do their job, they're going to be working in your capillary beds, like a big artery, like your aorta, or like the big veins you can see in your hand. Blood is not exchanging oxygen there. That's not the way this works. Oxygen exchange takes place in very, very small capillaries, where the red blood cells basically will line up and flow single file through those capillary beds to get where they're going, so that they can have very close contact with the surrounding tissues and thus release oxygen and take up excess carbon dioxide. So the primary functions of our red blood cells, let's see, good. Oh, there's a few more things to talk about here. The primary functions of our red blood cells is to carry oxygen from the lungs to the tissues, absolutely factual. They will pick up a small amount of carbon dioxide and carry that back to the lungs. Again, most of the carbon dioxide is carried in the plasma. And you need to consider that at the end of the day, a red blood cell is basically just a bag of hemoglobin, all right? The red blood cell contains a lot of water, but if you eliminate the water and just look at the actual components of the cell, it's almost entirely hemoglobin. And that's very important because hemoglobin is the oxygen-carrying device of your body, okay? What makes red blood cells red, that's the hemoglobin. Now, I neglected to point out one more thing that makes these wonderful oxygen-carrying tools. Your red blood cells still have a need for ATP, kind of to power some ion pumps and things to keep their ionic balance appropriate. And they do this, they make the ATP they need via anaerobic processes like glycolysis. They don't actually absorb any oxygen they're carrying, so they don't do any aerobic cellular respiration. They will keep all the oxygen for other tissues. They make the ATP they need anaerobically, which is amazing. It's pretty cool. All right, great. Erythrocytes and hemoglobin. So let's talk a little bit about that hemoglobin. Now, the first thing I want to say here is that hemoglobin, this is a small quaternary protein structure. Okay, a very small quaternary protein structure by comparison to the size of a red blood cell. I've even got some numbers for you here. One hemoglobin molecule can carry 4 individual oxygen molecules. One red blood cell contains 250 million hemoglobin molecules. What that means is a red blood cell can bank about a billion, if you will, a billion oxygen molecules. So these are very small. You're going to have 250 million of these hemoglobin molecules, again, a quaternary protein, 1, 2, 3, 4 structures, quaternary, in a single red blood cell, All right, so let's talk a little bit about hemoglobin itself. We've already done this. Let's go here. Actually, I tell you what, let's go here. Hemoglobin gives blood its red color. Okay, it just does. It's got a lot of iron in it, and when iron oxidizes, which is the nature of hemoglobin, is it's capable of taking iron and using it to bind oxygen, it gives blood this red coloration. And that shouldn't be a surprise to you because you've probably seen rusty iron before. Okay, if you haven't, here it is, and it takes on this kind of orangey red color. It's very much like your blood. The reason your blood's red is because there's iron in it, and that iron is binding oxygen. The reason it can change color is if it contains oxygen, again being a very bright red, or if it's devoid or very low in oxygen, it will take on a very deep crimsony red by comparison. So that would be very oxic, iron oxide. Let's see, what do we want to say? All right, I think we were talking about hemoglobin. So the nature of hemoglobin as a quaternary protein is that each of the polypeptide chains binds itself to a heme group. These heme groups have this beautiful ring formation, and like a jewel in the middle is this iron molecule. So there's an iron in the core of the heme, and then the heme is bound in a polypeptide, and there's 4 of these in a hemoglobin. So 1, 2, 3, 4 quaternary protein, 1, 2, 3, 4 heme groups, all bearing a single iron atom, which will bind reversibly to oxygen, which is great. This is how they carry oxygen. Again, what color is blood? It's red. It changes as a result of oxygen content. Carbon dioxide can be carried in small amounts in a red blood cell. No. Yeah, that'll do. In small amounts here, but most of it's carried in other ways. Perfect. All right, how is blood produced? And the answer is via a process we call hematopoiesis. Okay, hematopoiesis is the production of blood cells. And this is all going to be done in your red bone marrow. Now, when you're very young, you have lots and lots of red bone marrow because as you're growing, you're producing lots of red blood cells, as an example, or just blood cells generally. But you got to imagine the demand for the production of blood cells is very high for an infant because they're like doubling in size every year. Whereas you, as an adult, you have very little red bone marrow by comparison. Most of it is in your trabecular cavities, like in your pelvis and your sternum, for example. So that is the location of your red bone marrow where you're going to be doing what we call hematopoiesis. You, as an adult, will produce about 400 billion platelets a day, 200 billion red blood cells, and 10 billion white blood cells. So again, the platelets are fragments. 200 billion red blood cells is a big number. And again, think about that buffy coat, less than 1%. Very few white blood cells by comparison. Okay, very few. And what this amounts to is about an ounce of new blood produced in a day. And again, the process is hematopoiesis. It is done by your red bone marrow. Now, what we have in our red bone marrow are stem cells, and we're going to be talking all about these as we progress. But the idea is that we have very special stem cells located in our red bone marrow, and they can produce precursor cells that will lead to a variety of different cell types. All right. So stem cells, still in your system in a stem state, they will produce various different precursor cells that will then lead to the major cell types that we think of when we think about the blood. And that's what we're getting at here. So let's go through and have a little conversation on the subject. Let's talk about erythrocyte production, what's referred to as erythropoiesis. All right, a few things to discuss. We've got a hematopoietic stem cell, as we were just discussing on the previous slide. These will be referred to oftentimes as a hemocytoblast. A hemocytoblast. Okay, hematopoietic stem cell, the stem cell from which red blood cells and white blood cells, what have you, are produced, oftentimes called hemocytoblasts. And again, they lead to all the different major cell types that we find in the blood. What will happen is this will release a cell that differentiates into what we call a pro-erythroblast. Okay, pro, we're before making a erythrocyte. Erythroblast. Blast is we're making something. Erythro, we're talking about red blood cells. Pro, we're before we're making red blood cells. So it's an early, what we call a colony forming cell. This is what we'd refer to as being committed. Okay, this is an important term. When a cell is committed, that means it's going down a certain street. Imagine a fork in the road, and it's picked that way. What it's doing is it's heading towards being a red blood cell. Pro-erythroblasts, we're headed towards being a red blood cell. It's going to be an erythroblast eventually. It's going to eventually become a reticulocyte. And then that will mature, if you will, into an erythrocyte. So we have a hematopoietic stem cell, oftentimes referred to as a hemocytoblast, that will release a cell that will differentiate into a pro-erythroblast, which is what we refer to as being committed. Okay, it's committed to becoming a red blood cell, and then it's going to run down a list. Now, An important nuance to this, and the reason we're really talking about this, is because pro-erythroblasts respond to a hormone called erythropoietin. Okay, they respond to erythropoietin. Erythropoietin is released by the kidneys through a process I'll show you soon, and basically stimulates for red blood cell formation. So these will hang out, and when they start getting erythropoietin, they're going to start cranking through and producing red blood cells. All right, you're going to be making red blood cells like it's going out of style when you've got appropriate amounts of erythropoietin in your system. Now, you have a base level of erythropoietin so that you're constantly making that small amount of red blood cells that we were talking about previously. But if you were to become injured, or if you were to go into a low oxygen environment, you'd start producing lots more erythropoietin. So that would stimulate these pro-erythroblasts to develop very quickly. All right, so that's very important. These are colony-forming cells. They are committed, pro-erythroblasts are committed to becoming red blood cells, and they respond to erythropoietin. Very important. Erythroblasts, what makes them kind of unique is they have huge numbers of ribosomes. Again, ribosomes are the site of protein synthesis, and what's hemoglobin made out of, right? We're going to be packing that cell with hemoglobin, and hemoglobin is a protein structure. So we got to make all the protein in the world, so this thing's got to be packed with ribosomes, and that's its main facet. Next, along the path of development are the reticulocytes. They will basically have shed their nucleus, shed most of their major organelles, and they will get that biconcave shape here. It still will have some reticulum, the endoplasmic reticulum and ribosomes. We're still making a little bit of hemoglobin here, and that's why it's called a reticulocyte, because it's pretty much just got endoplasmic reticulum left. And it takes, from the origin of this process, about 15 days to get to this state. And then after a couple of days in the bloodstream, those are going to mature into true erythrocytes and be capable of doing the job of red blood cells. And did I mention this? Yeah, over here. So reticulocytes, we can actually count these, and it gives us a good grasp of how many red blood cells are being produced by the body in a given period of time. So by looking at the number of erythrocytes, we know how long it takes to get there. We know how long they stay in flow before they mature into red blood cells. By counting the number of reticulocytes, we can get a good idea of the health of the system in terms of producing red blood cells. So it's a very important determinant in certain medical conditions. All right, so we know about the formed elements. We know about red blood cells. We know kind of what they do. How do we keep their numbers relatively constant? Okay, how do they vary over time? This is a very important process, and it's going to involve that hormone erythropoietin that we talked about a couple minutes ago. It's very important to maintain erythrocyte count. What this is going to do is we have to have enough red blood cells that we can carry oxygen effectively. If the number of red blood cells drop, you can find yourself having a hypoxic environment. You can have not enough oxygen carrying capacity, and if your oxygen carrying capacity drops, you're going to feel fatigued all the time. You're going to be tired, like just getting up, you're going to be so worn out. That's because you don't have enough red blood cells, okay? Alternatively, if you're producing too many red blood cells, you can carry oxygen like a champ, but the blood will get too thick. The viscosity goes haywire. And if the viscosity goes crazy, it can lead to the blood being too thick. It's too hard to pump the blood, so your blood pressure rises. You can have a stroke, you can have a heart attack. It can all get all clumped up and just turn to molasses. That's a bad move, okay, real mistake. So we want to prevent that. So how do we do it? Well, it's a negative feedback control system, as with every homeostatic function of the body. For the most part, these are all going to be negative feedback systems, and it goes as follows. If you have a drop in red blood cell count, that's going to make the kidneys go hypoxic. They're going to be in a low oxygen environment. And when the kidneys find themselves in a low oxygen environment, they don't care for that at all. So they release erythropoietin. And erythropoietin goes into the bloodstream, flows around until it finds your bone marrow, and stimulates your bone marrow to produce red blood cells. And it won't take long either. It'll just take a few days for the erythropoietin to really start cranking on those progenitor cells and get them to be producing red blood cells. So it's a pretty quick process, just a few days. So what causes this? Again, low oxygen environments. If you find yourself up on top of a mountain or something like that, like at high altitude, that can certainly drive red blood cell production. Increasing in exercise. So if you're working out all the time, jogging, running, swimming, what have you, if you're really working out, that means you're using a lot of oxygen constantly, and your kidneys are going to pick up that low oxygen environment and be like, hey man, we need more red blood cells. They're going to start cranking out erythropoietin. You can, emphysema. So if your lungs aren't being efficient as a result of some lung tissue being destroyed, that means that your kidneys are going to sense a low oxygen environment, so they're going to release erythropoietin and up your red blood cell count. And again, testosterone. So testosterone has shown to lead the kidneys to releasing more erythropoietin, which means more red blood cells, which would account for the variation in hematocrit between males and females. Okay, and I can't just leave this be without talking about top-tier athletes and the way that they train. So you probably have heard of high altitude training. There are, famous stories, go watch Rocky or something along those lines, of these folks that will want to be more athletically capable in a certain time period. So they'll go and they'll start training in the mountains for a few weeks or a month prior to some sort of event. And what that's going to do is it's going to put them in a low oxygen environment, lead them to produce more erythropoietin, and lead them to have more red blood cells in a relatively short period of time. And unfortunately, and I may have this in here someplace, I honestly forget, this also leads to another sort of dirty underbelly in the sports world, referred to as blood doping. He's kind of an old hat these days, but you guys may have heard of Lance Armstrong. Lance Armstrong won the Tour de France several times. He was a dominant cyclist, just absolutely dominant. And it turns out there was a reason he was absolutely dominant. He was blood doping the whole time. So he was taking, I believe, artificial erythropoietin injections. Okay, he was injecting himself with erythropoietin, which was upping his red blood cell count and giving him more oxygen carrying capacity, and thus an increased ability for athletic behavior, if you will, aerobic respiration. And then there is even another way that some folks will do this. Again, red blood cells last for a period of time. There are situations where athletes have gone and had a certain amount of blood pulled from their system, like for instance a pint. They'll drain a pint of blood out of their system and put it on a cool, and then they will exercise and do what they do for a few weeks, and their red blood cell count will get back to normal. And then prior to an athletic event, they'll take the blood they drew out and re-inject it, which dramatically increases their red blood cell count. And it's real dangerous. It's real dangerous. Go and look it up. People have died doing this because their blood gets too thick, and then they're engaging in some sort of athletic activity, and they have a heart attack or a stroke. Bodies can't take it. The cardiovascular network can't handle it. So it's very important. Okay, this is an important thing to point out for us. The kidneys play a good role here in the regulation of our red blood cells. Next, I want to talk just very briefly about the dietary requirements for erythropoiesis. The idea here is, again, if we're making red blood cells, you can imagine we need a lot of iron in the diet. So you got to be bringing in plenty of iron if you're going to be making all of this hemoglobin, for example. So plenty of iron, plenty of amino acids for the production of those polypeptide chains. Certain lipids and carbohydrates are also going to be involved here. And an important part of this is B12 complex vitamins. Okay, or B complex vitamins like B12 and folic acid. These are very important for making cells quickly. If you're trying to go mitotic and really divide cells fast, you gotta have these or it's just not gonna work. They're involved in DNA synthesis. So they are very important. So if you look at, for instance, a good example of this is all right, folks, we are changing scenery yet again. This first lecture is proving to be lots of fun to get recorded. So now we are in my office, and we are shooting just the screen of my computer, and we're going to go from there. I believe we were on this slide, and we had established that you need certain things for general dietary intake in order to make red blood cells, like iron, for example, folic acid constituents. These are very important for general synthesis of cells as they go. And I've included this text below. I'm not going to go into a huge amount of detail on it, It is important, but it's not anything I'm going to test on, so we're just going to sort of briefly touch on just a couple of concepts. And what I want to touch in is the nature of ferritin, apoferritin, transferrin. Anytime you see any of these, gastroferritin, all right, all these, anything ferrite, anything you see, any of these, you got to understand that these are going to be transporting or be part and parcel with iron. Iron is dangerous to your system in its raw form. Raw iron, as you can see here, these are not ideal to be just flowing through your system. They have to be bound up in a specific way to move through your system in a non-toxic fashion, and then whenever you relieve them from your system, as you'll see in a second, they have to be relieved in a very specific way as well. So this is the death of erythrocytes and sort of their disposal. Again, your red blood cells contain a lot of iron, right? And that iron is toxic, so we have to get rid of it in a very specific way. All right, so let's run through this. As your red blood cells age, remember they last, somewhere in the nature of 120 days, they will eventually, as a result of not having a nucleus and not really being able to repair themselves if they're damaged, they'll become too rigid, they don't flex enough, and they get fragile. So they begin to fragment, in essence. They don't function well, and so your body's got to get rid of them. So how do we get rid of them? By the way, small intestine, these are important nutrients for you to take in going into the bone marrow to make red blood cells. Those red blood cells get old, and then we have to get rid of them. Now, this is done in two organs primarily, the liver and the spleen, but you need to know that the spleen runs the show here, team. The spleen is most important. The spleen is the organ that we use primarily for the breakdown of red blood cells. And what will happen is, red blood cells, when they get old and they lose their flexibility, they get into the spleen, they get kind of caught in the matrix of the spleen, and then broken down by macrophages. All right, this is sort of the way it happens. They get into the spleen when they're old, they can't move around as well as they should, so they get trapped in the spleen, and macrophages in the spleen destroy them. Now, the hemoglobin itself is broken down. The globin, the protein network itself, gets broken down into individual amino acids and just dumped right back into the body to be used. Okay, amino acids are always important for us, so it's not a big deal at all. We'll reuse those amino acids like it's going out of style. But the iron constituent from the heme, all right, it has to be removed in a very specific way. All right, as it comes out, it's coming out in a format of bilirubin. Okay, bilirubin is secreted in bile via your liver, and that's going to be part and parcel with your digestive system. Okay, so the bile is going to contain this bilirubin. It's going to go through a few other changes as it moves through your digestive tract, and by the way, the bile is going to help you absorb lipids. It's sort of a neat little twist to this, but regardless of that, the bilirubin is going to be broken down and eventually turned to a brownish pigment and released via your feces. In fact, the reason feces have a brownish color is as a result of, as a trip up the line here, this bilirubin. Okay, it's a major part of this. It's in essence an iron constituent. So you're getting that brownish, rusty color in the grand scheme. Now, bilirubin itself is a yellow pigment. It's broken down into this brown pigment that we're talking about. And it's kind of important to go ahead and mention jaundice. So jaundice is a liver condition. Jaundice itself, a yellowing of the skin, yellowing of the eyes, is not a medical condition. It is a sign that you have a liver issue for the most part. All right, when your liver is not appropriately utilizing bilirubin, and it can build up in your bloodstream and thus build up in your tissues and lead to jaundice. All right. Or in the case of very young children, like newborn babies, oftentimes you see a lot of newborns born with jaundice. It's just a factor of the liver not yet working as well as it should. So you give the body a little bit of time, it'll catch right back up. All right, anemias. That brings us up to the concept of anemia. So anemia in the grand scheme is a decrease in the body's blood oxygen carrying capacity. Okay, this could be caused by just, a whole host of things. This is yet another example of a condition of a disorder. Anemia is not a disorder. Anemia is a side effect of something else completely different happening. You see a lot of this in kidney failure. People that have dietary issues, like, just be frank with you, some you see this periodically with vegetarians, all right? Folks that aren't eating enough meat, they're not getting a lot of iron, so they can end up with some degree of anemia as a result of that. Simply, the blood is losing its oxygen carrying capacity. Now, there are three main categories that I expect you to be friends with. These are hemorrhagic anemia. That's a result of blood loss. Imagine you get a deep stab wound or something, you bleed out a lot. That's going to decrease your body's oxygen carrying capacity. There's also chronic versions of this, where somebody has like an ulcer or something that's constantly bleeding, or some sort of intestinal issue. They're going to be constantly losing blood, so they're going to be anemic. Again, they'll be tired. They'll have a lot of fatigue. These are classic examples of anemic issues, if you will. We have inadequate erythropoiesis. Okay, this is typically of not having enough iron or not being able to absorb iron correctly. This is also part and parcel with your urinary system. So if your kidneys are not releasing appropriate amounts of erythropoietin, that could be inadequate erythropoiesis. That's another form of anemia. Now, there are two nutrient-based anemias. Again, think about dietary deficiencies. Iron-deficient anemia, where you're simply not ingesting enough or not absorbing it for one reason or another. And then kind of famous is pernicious anemia. This is a problem with B12's absorption, okay? When you're having issues with B12, that's referred to as pernicious anemia. Yeah, that'll do. That'll do. Again, B12, very important in the process of forming those red blood cells, and particularly that hemoglobin. All right, and then last but not least is hemolytic anemia. Hemolytic anemia is where your erythrocytes are rupturing prematurely. This can be due to a whole host of things, bacteria, parasitic infections, but also falling into the realm of hemolytic anemias are sickle cell and the thalassemias, okay? Sickle cell kind of famously in this. This is going to be a condition where the red blood cells can oftentimes form up improperly. And you can imagine that these cells are not going to work as well as they should. They can sort of clog up capillary beds. They can break open when they're not supposed to. Classic hemolytic-based anemia. Okay. Consequences of anemia are pretty obvious, and we sort of already talked about this, but I'll hit it one more time just because. Tissue hypoxia, so your tissues are not getting enough oxygen. Pale skin is a sign of this. Shortness of breath, you hurt. It's painful when your tissues aren't getting enough oxygen. Reduced blood osmolarity. When you play with the blood's osmotic balance, you end up releasing entirely more fluid from the bloodstream than you should, and this ends up causing localized swelling in a lot of cases, or even body-wide in certain medical conditions, which is not ideal. And then, of course, there's reduced blood viscosity. So when you're anemic, your blood loses viscosity, so it's more watery, so it flows through your bloodstream very fast and with low resistance, and the heart doesn't do well in that situation. You need a certain level of blood viscosity for the heart to be able to pump correctly, and this drops your blood pressure, which, again, if you've ever stood up quickly and then felt kind of woozy, that's a classic low blood pressure situation. So the heart isn't being very efficient with pumping blood, and that causes the heart rate to go up, trying to keep up with the oxygen demands. It's just very unhealthy for your cardiovascular network. Yeah, that'll do. All right, let's move on to something a little different. Now, your book puts this in a slightly different place, but because we are talking about red blood cells, I feel like it's a good time to go ahead and talk about blood typing, because blood types depend upon your red blood cells. All right, very important in this concept. So what are we going to do? We're going to go through and talk about the classic ABO and RH blood types. That would be positive and negative for those of you wondering. So you probably know your blood type, or you may have heard someone in your family say they're A positive, or they're AB negative, or O positive, anything along those lines. We're talking blood types here. And I want to talk a little bit about an interesting nuance to this, which deals with a mother and her offspring. Kind of a neat thing. So we'll get there in a second. It's referred to as hemolytic disease of newborns. And again, this is not a problem. It sounds terrifying, but not a problem. We take care of this pretty easy these days. All right, so this is a slide you have seen if you've ever taken a class with me before. You know that you have a cell membrane on all of your cells, And those cell membranes have a variety of structures. For instance, we have these glycoproteins and glycolipids on the outside of your cells. These are sugar coatings, if you will, referred to as your glycocalyx. And this matters to us because our blood types, all right, our blood types are based off of the sugar coating, if you will, on the outside of our red blood cells. So our red blood cells, they have markers on their external surface, and those markers give rise to our A, B, and O blood types, as well as being positive or negative for that rhesus factor. So blood types are important because this plays into the concept of transfusions, plays into the concept of doing transplantation of organs. It's going to be very important in and of this. We didn't always know about blood types, and that obviously gave rise to some problems in the past, but this really was discovered around 1900, and the guy that did it, that made the discovery, was given a Nobel Prize in the 30s, so that's great. Okay, that's great. So, why do a blood transfusion? That's the first question. When you lose up to about 30% of your blood complement, you get very weak, pallor, your skin coloration changes, and you are experiencing that anemic feeling, if you will. And once you pass about 30%, you go into what's called hydrostatic shock. Again, we are back to here with low blood viscosity, resulting in all kind of cardiac issues. Okay, so this is dangerous. Having low blood content, losing a lot of blood is very dangerous. So what do? We will pipe in fresh to you. Yeah, that's what I'm getting at here. So when we give you whole blood, that'd be red blood cells, plasma, and platelets. When you get a whole blood transfusion, you can't just pipe in whatever the heck you want and call it a day. That is not the way this works. We have to be mindful of the interactions between what are called antigens and antibodies. Antigens and antibodies. So what are these? Okay, well, antigens are surface markers, if you will, and there's a lot of these. So when you have a very fancy network that you're going to hear all about later called your major histocompatibility complex, and this would include part and parcel with lots of different antigens. We are going to be concerned with very few of these that are specifically referred to as the agglutinogens. These are antigens on the surfaces of our red blood cells that are the basis of blood typing because they cause agglutination. Agglutination is the clumping, if you will, of blood given certain circumstances. So antigens are on the surface of your red blood cells. They are surface markers for your red blood cells. Antigens are surface markers. So what are antibodies? Immunoglobulins, okay? Antibodies are part of your immune system. They're flowing around in your serum. These tiny little markers is what they are. Tiny little antibodies floating around in your blood plasma. And what they will do is they can mark a cell and cause it to be destroyed. They mark them for death. Okay, that's what antigens do. Antigens are part of the immune system. They mark foreign cells for death. All of your cells have markers on their external surface that say that they belong to you. If your antibodies pick up antigens that don't belong to you, they mark them, and those cells get killed, man. They're destroyed. Like this cell here. It's got surface markers. These are antigens. Surface markers, antigens. And these are antibodies that have picked up those surface markers, recognized this cell as being foreign, and marked it so a big macrophage can come through and nom destroy this thing. Okay? So antigens are surface markers. Antibodies are in your plasma. They are part of your immune system. Good. So what do these do? When these antibodies mark your red blood cells, they lead to clumping or agglutination, and clumping up of your red blood cells is very dangerous. So if you are type A blood and they give you type B whole blood, you're going to get all kinds of crazy blood clotting internally, and that can kill you real fast. So we don't want to do that. That is very bad. So what we need to do is understand the concept of blood typing and who gets what in terms of donors and recipients. And that takes us to this beautiful, nice slide. Okay, let's talk a little bit about blood typing. So here is a person with type A blood. here's a person with type B blood, here's a person with type AB blood, and here's a person with type O blood. You might be type A, type B, type AB, or type O. You could be any of these. But what does it mean to be type A blood? Man, let me tell you, people confuse this so many times. It seems like nobody gets it. But I don't grasp how it's possible to mix this up, because it's so simple. So I'm going to lay it out to you in that simple way. If you're type A blood, that means you got type A antigens. If you're type B blood, you got type B antigens. If you're type AB, your red blood cells have both A and B antigens. Not a big deal. You just got both. All right? Not a big deal. If you're type O blood, you have no antigens. Your red blood cells are naked. All right? They have no antigens on their external surface. So what does this mean? Well, a person that's type O blood, they have no markers on their red blood cells. They are considered a universal donor. That means that their red blood cells, you could pipe it into anybody. That person's going to be totally fine. Their body is incapable of realizing that someone else's blood is in them, because that someone else's blood has no markers on its external surface. They're ghost cells for your immune system. Your immune system can't detect these. They don't have any markers. A person that's type O can give blood to anyone. However, a person that's type O will have an immune reaction to any other red blood cell type that's put into their system. Now, what the heck am I talking about? Let's jump up to a simple example. A person's type A. That means that their red blood cells have type A antigens on their surface. Well, if they are given type B blood, their immune system will recognize that this is foreign. And when it recognizes that this is foreign, they will build type B antibodies. We call them anti-Bs, okay? So I'm type A. Somebody gives me type B blood, I'm going to make antibodies against it, mark those cells, and destroy them. I'm going to end up with all kinds of crazy agglutination taking place. If a person is type B blood, then they have the capacity to make type A antibodies. So if I'm type B, and you give me type A blood, I will make antibodies against that blood, and I will destroy it. If a person is type AB, that means they have both A antigens and B antigens on their red blood cells. Well, you can pipe in any kind of blood the heck you want to this person. They're going to be totally fine. They're a universal recipient because they already have A antigens. They already have B antigens. They don't make any antibodies against any of this. So you can give them any kind of blood the heck they want, and they're going to be totally fine. Whereas our universal donor here that can give blood to anybody, they have no markers on their red blood cells. So if they get any kind of blood other than type O, they'll make antibodies against it and lead to agglutination and problems. All right? So a person whom is type AB is a universal recipient that can get blood from anybody. A person that is type O is a universal donor. They can give blood to anybody. But the trick is, again, if I'm type A and you give me type B blood, I'm going to make antibodies against it. My immune system is going to destroy it. It will recognize that as foreign. It will recognize type B blood as foreign and destroy it, leading to clumping, leading to that agglutination. Yeah. It's just your immune system, folks. It's just how your immune system works. And that's going to take us to... And that takes us to the rhesus factor. So, rhesus factor dictates what's referred to as being positive or negative. This was discovered in rhesus macaques. It's a monkey, if you will. So we call it the RH factor or rhesus factor. This is just basically a variation on being positive or negative, okay? So you might be type A positive. That means you'd have type A antigens, and you're positive for this rhesus factor. It's just another surface marker, okay? Don't think of it as anything more complex. It's just another surface marker. And the one that there's several of these rhesus factors. There's a lot of different ones, but the one that leads to agglutination is rhesus D. So you'll oftentimes see this RHD positive or negative, okay? Normally it's kept RH positive or negative, but you may see the D. So I'm just telling you what that is. It's just the one that leads to agglutination. Now, this is interesting mainly for one reason, and that is dealing with pregnancy. Okay, you may have heard that in the past people had to get a blood test when they were getting married, and one of the reasons for this is to see if they may end up with a problem dealing with their offspring. Now, let me explain what the heck's happening here. If mom is rhesus negative and dad is rhesus positive, that means that the offspring that they might eventually have during development in utero could potentially be rhesus positive. Okay, depends on dad's genetics, but if dad's rhesus positive, the kid might be rhesus positive, and if the kid's rhesus positive, what will happen is when the child is born, at the placenta in particular, there would be a lot of mingling of mom's blood and the baby's blood. Okay, it's going to mingle together. And when that happens, mom's immune system is going to recognize this rhesus positive blood and build antibodies. Not a big deal. This is just your immune system working, all right? Your immune system, when it detects foreign cells, it's going to build antibodies against them. So when the kid's born, mom is going to have rhesus positive blood in her bloodstream. Her immune system is going to detect this and destroy those rhesus positive cells by building antibodies that mark them for death. Not a big deal. One kid, not even remotely a problem. But now, mom has antibodies against rhesus positive blood. And if mom's got antibodies against rhesus positive blood, that can be a problem with the next kid. Child 2. If child 2 is rhesus positive, mom's antibodies will cross the placenta and lead to major damage to that kid, all right? Lead to problems. Because mom's antibodies are going to bind to the kid's developing red blood cells and mark them for death, and the kid's immune system is going to fight them, okay? It's going to start having macrophages attack and destroy developing red blood cells. This is not good, okay? This is not good. And in the past, it's been a real problem, but no longer, because today, what we do is if mom is rhesus negative and the kid potentially is rhesus positive, what we'll do is we'll give mom, right when she gives birth, we'll give her a shot of what's called Rogam. Okay, the kid's out of her system, we'll give her a shot of Rogam. And what this is rhesus positive antibodies. What we're doing is, instead of letting mom, let me try again in English. Instead of letting mom's immune system fight off this rhesus positive blood, we are giving her antibodies that do the fighting for her immune system, so that her immune system stays what we call naive. That way, mom doesn't develop antibodies, so the next kid will have no problems. So let's run through this one more time. The kid's rhesus positive. When mom gives birth, there can be a mingling of the blood, and mom will develop antibodies against rhesus positive blood. To prevent this, we give her a shot called Rogam shortly after birth, and this shot contains rhesus positive antibodies, and it will go through and destroy any rhesus positive blood that makes its way into her system without her immune system having to lift a finger. Basically keeping her immune system naive so that she doesn't develop problems with any further offspring that she has. This is referred to as being hemolytic disease of newborns and is easily fixed using this shot. Yeah. All right, so this has been the first 20 slides. Let's just leave it there. I want to get something up to you guys, so let's just go ahead and get this uploaded, and you can expect the next half of this very soon. All right, thanks guys. Have a good one.